This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 581, recorded on January 3rd, 2020. Happy New Year, everybody. I'm Vincent Racaniello, and you are listening to the podcast all about viruses. Joining me today here in New York City, Dickson, Despommier.
Hello, Vincent.
Not a nice day, is it?
It's yucky out. Yeah, it's cold and wet.
Yeah, it's kind of raining. And
but it wasn't cold and raining where I got back from just recently.
8 degrees Celsius.
Got back from Morocco.
You were in Morocco. We're going to see where people were on this podcast.
Yeah, okay. I'll just throw my hat in there.
Morocco, and you brought some dates in today.
I did.
All right. Also joining us from Western Massachusetts, Alan Dove.
Good to be here. And it's almost the same weather here. We have 8 Celsius, 47 Fahrenheit, and cloudy and blech.
Have you had any snow?
Yeah, we had snow and we had an ice storm. And it's been a weird winter. And now it's above freezing and everything's melting and muddy. And then we're supposed to get a little more snow. So it's just kind of back and forth.
Also joining us from Madison, New Jersey, Brienne Barker.
Hey, it's great to be here. After, seems like a long time away.
Yeah, it seems like a long time.
So it's sunny and 80 degrees where you are? Is that right?
No, strangely enough, it's the same weather here.
Get out of here. How
with
could that
you
be?
and with Alan.
Oh, my gosh.
Yeah, we did. What was our last TWiV? Let's take a look here.
It was a while back.
It was
in December.
Yes, it was December. Here we go. It was 573 is November. It was Bill Summers. And then after that, every episode, as I'm looking through here, was on the road.
I'll be darned.
It's been a long time, yeah.
Also joining us from southeastern Michigan, Kathy Spindler.
Hi, everybody. Here, It's 41 degrees Fahrenheit. I don't have an easy way to do Celsius anymore, but yeah, it's just gray, but it's not bad. And while I was gone, I was in Yellowstone and Tetons.
Nice.
For Christmas.
Fabulous,
and it's wonderful in the winter. If you've never been to Yellowstone, winter would be a good time to go because there's fewer people. And if you've been in the summer, winter's a good time to go because there's fewer people. And it's just magical to see all the thermal features in the snow. And we're also in the Tetons. So mostly getting around by snowshoeing.
Did you see any wolves?
We saw wolf tracks.
Excellent.
In 2 or 3 different places. 3 different places.
Fantastic.
And snowshoe hare tracks, which were the best.
Oh, great.
Yeah.
Alan, did you go somewhere?
No, didn't go anywhere interesting.
Brienne, how about you?
I went to my family's in upstate New York.
Right.
Yeah, I didn't go anywhere over the holidays, actually. It was here. But I did early in the first week in December. I did go to Singapore, which was fun. Well, it was a very informative trip because I didn't do anything. Everyone told me you should do this and that, so I didn't. I went to a meeting and then went to 2 places, and so in total I have 4 TWiVs from Singapore, which will be coming up in the near future. It was a lot of fun for science. I mean, the NEPA meeting was great. And then I visited the National University of Singapore Duke Medical School for one day, and then I went to their CDC equivalent another day. And as I was telling Dickson, I visited a facility where they make Wolbachia mosquitoes for release. They have a huge release program. And when I released that episode, I interviewed the director of environmental health or something, and I will talk about that in more detail. They have an amazing production facility. It had just opened, even though they'd been making many millions of mosquitoes for a long time. So that'll be cool to talk about. I was very impressed. Well, anyway, Happy New Year, listeners. If you like what we do, think about supporting us financially in this new year, in the new decade, microbe.tv/contribute. Let's start with some announcements from Kathy Spindler.
Well, since you've been on the road so much, we haven't had any of our priming announcements about ASV annual meeting that we try to do in December. The meeting is coming up in June. It's earlier this year than usual, June 13th to 17th. And that means that it's time now to pay your membership dues if you're behind. And if you're a trainee and you're going to submit a travel award application, you need to be a member. So you need to get your membership up to date now. And the abstract deadline is a day earlier than usual. It's January 31st. So this meeting in Colorado State at Fort Collins will be a great meeting, as they all are. And just a couple of other things over the next couple of weeks, we'll tell you more about some of the special features of this meeting. But we have a new award this year, the Global Virology Scholar. And that travel award has an earlier application deadline than anything else. That's June 13th. So we used to have Latin American scholars, and we've expanded it now to Global Virology Scholars. So if you were planning to do a Latin American Scholar Award, or if you're from elsewhere around the globe, January 13th is your date. So we also have a thing of, we instituted early decision for abstracts, where if you're going to need to get a visa, you can submit your abstract now and find out at least that it's accepted. And then that'll allow you to apply for your visa right away. You won't find out whether it's accepted for oral or poster presentation until the same date as everyone else later. But we have this early decision mechanism, which you'll see as you submit your abstract. So any kind of details that you need, go to asv2020.com, and that will give you all the details you need.
So with all that, is it safe to say that it's easier than ever to go to ASV?
Should be. Yeah.
Interesting logo for Fort Collins this year. Very cool.
Mm-hmm.
Very cool.
Yeah.
I like that. So if you go to the website, you can tell if you've paid your dues this year, because that's where I am right now. And it says that I have paid.
Whew, good.
What is it? I guess CC means credit card, right?
Yep.
Yeah, I paid.
I'm not seeing the logo.
ASV2020.com.
ASV. Oh, I've got.
Interestingly, it's at the top of the renew dues page.
Oh, there it is.
There's a
Cool.
picture of the logo as well.
Yeah.
It looks like a melting adenovirus.
Yeah.
Kind of.
Rising over the mountains.
Yep.
And it's the Colorado flag.
Yeah.
Oh.
How'd that-
Okay.
Yeah, you got me there.
And the actual mountain formation is real. You can see it from Fort Collins. Jeff Willis told me what it is, but I don't remember at the moment.
Got it.
It's not Pikes Peak, I don't think.
No.
Fort Collins is
is
also the home of WWV radio station.
It's
for anybody
also
who
the
cares.
home of the National Arbovirus Laboratories.
Mm-hmm.
WWV has the most regular and boring broadcasts of any radio station in the world.
Really?
Oh.
It's the time.
Oh.
That's the broadcast of the atomic clock time from National Institute of Standards.
How often do they say it? Every second?
Every minute.
Wow.
But it has a whole, there's this whole encoded thing to it. It's got a tick exactly every second, and there's a tone, and the tone is exactly a particular frequency, and the frequency on which they transmit is exactly, they're at 5 megahertz, 10 megahertz, 15 megahertz. You can set a frequency calibration to them. You can standardize all kinds of things to WWV.
Is that a hard-bodied tick or a soft-bodied?
It's grown in a tickery.
And
Tickery.
Is that the National Center for Standards also?
National Institute for Standards, yeah.
Yep, okay.
Tickery dock.
Yes.
Dock. So Fort Collins is very nice. For those of you thinking are on the fence, you should go because it's very interesting.
Except when they have fires.
Yes. And there's
And they often
just
do.
right after ASV, there's a bat meeting,
Really?
which I'm going to go to.
Huh.
And they invited me, and maybe I'll do another TWiV with some bats.
Which ball field are they having that
I don't know. Going to go to bat for the bats. And the town is wonderful. Little town, lots of micro brews. Really cool. Looking forward to it. I want to tell you a norovirus story.
Uh-oh.
Pull up a chair, kids.
No, it's not going to be long.
Oh.
No, it's not
Uncle
a chair.
Vincent's going to tell us
No,
this.
so my wife had surgery mid-December, and she had abdominal surgery, right? She had a piece of her colon removed. Everything's good now, But the morning after, I had left her in the evening of surgery in her room, and the person next to her left. And then the next morning, she said they put another person in who had a raging norovirus infection, a 92-year-old woman who was having vomiting and diarrhea. I said, that's not good. It can aerosolize, And she said, yeah, I said to the nurses, can you get me out of here? And they said, well, we wash our hands.
Do you wash the air?
So infection control is not what it should be. Fortunately, she didn't get an infection, which would have been bad with the suture in the colon, right?
Wow.
Yeah. Right.
So she was fine. And
they should have at least given her like a mask or something. Wow.
And then they moved her. She said, well, this is happening at all hours, right? So people were yelling and screaming. And at one point, the woman said, I have to go. And the nurse said, sorry, there are only 2 of us on the whole floor. Just go in your diaper, right? With norovirus. How bad. So anyway, they moved my wife to another room. And then there was a guy who pulled his IV out every hour. And the nurse came running in, yelling at him.
Which hospital was this? Which asylum was that at?
It's a good hospital. They have lots of Columbia adjuncts there. Anyway, that's norovirus.
Woof.
The other thing I wanted to point out is an article in Science magazine, and this is the Italian scientist who has become a celebrity by fighting vaccine skeptics. We mentioned this before last year. Someone sent it in to us, but this is a lovely article. The guy, his name is Roberto Burioni. He's a virologist at Vita Salute San Raffaele University Milano. And he is speaking out against anti-vaxxers. It's great. He's become incredibly popular. And he has this quote. He went on to this talk show. And he had a few minutes to give good arguments for vaccines. So he just said, the earth is round, gasoline is flammable, and vaccines are safe and effective. All the rest are dangerous lies. And apparently now he's like incredibly famous, and he's pushing vaccination, and it's great. And someone said here, he's one scientist, Guido Silvestri from Emory. He's one scientist who stood up and said, this is bullshit. Right? It's true.
Yeah, they put him. So they put him on a show with a couple of celebrities who were anti-vaccine, and they gave most of the airtime to the celebrities. And then the last couple of minutes, they said, turn to the scientist to have the token opinion in there, and this is the way he put it, and it's
great.
Well put.
It's great.
You bet.
So it is. It's all BS. Everything else is BS, and we can spend hours and days and weeks telling you why, but it's all BS.
Yeah.
So thank you, Roberto Burioni. This is great. I mean, we try and do this here in a more reserved academic way, but maybe it doesn't work. We actually have an email, which we'll look at next time, where the guy basically said, you guys say this anti-vax stuff is BS, but we want you to explain why. I don't know. Maybe we should just say it's a BS.
Yeah.
It worked for Roberto. Okay, we have 2 interesting papers to bring us back into the TWiV fold. I'm sorry that Rich is not here, but he will be here next year. And the first is
next
wait, whoa.
next week. Sometimes I'm
okay.
glad you're listening. I'm glad you're listening. First is a paper. Our snippet is a paper published in Cell, which I blogged about some time ago, and I thought it was important for our TWiV listeners to hear about. It is called Antibiotics-Driven Gut Microbiome Perturbation Alters Immunity to Vaccines in Humans. A very large cast from Stanford and University of Chicago and the Racon Institute, Georgia State, FDA, University of São Paulo, Institut Pasteur. We have, we have 3 co-lead authors, Thomas Hagen, Mario Cortese, Nadine Rufael, and then we have a lead author, Bali Pulendran. And so yesterday on TWiV, we did a microbiome story, very interesting, where, certain microbiomes track with colon cancer. And very interesting story there. Here, there's some evidence that the gut microbiome in particular, which, as we have heard in the past few years, apparently controls everything. From health to illness.
This is true.
It controls the stock market.
I think I saw something in recent days that this had been the decade of the microbiome.
Yep.
I thought that was the next decade. That's the decade we're in now, right? Yeah, this is the coming 2020. Did I say that 2020 is a great year?
It will be.
I mean, it's even. It sounds great, 2020. And just think 3030 is going to be the next one. And I'm not going to be here.
Oh, I've thought about that.
None of us are going to be here.
You won't even be here for the palindromic equivalent.
The last one, 1010, was kind of screwed up, right?
1010 was really
a lot of wars going on.
Shirley MacLaine might be here in 3030.
It was the Dark Ages.
Some version of her.
2020 is
some
great.
Version.
Anyway,
right.
there's some evidence in mouse models that gut microbiome can influence responses to vaccination. In particular, there's some work by this group in particular. If you take TLR5, toll-like receptor 5 knockout mice, which can't sense flagellin, component of many bacteria, these mice have impaired responses to inactivated influenza vaccination. Of course, that's mice. So what about people? And they could actually do an experiment in people, which is neat.
Yes.
Because you can give people antibiotics, because we do it all the time. So they decided,
often when they don't even need them. So yeah,
a natural experiment.
So this was done in the 2014-15 flu season. They took 22 healthy individuals, 18 to 45 years of age, and they gave them a 5-day broad-spectrum antibiotic regimen. Neomycin, vancomycin. Sorry, half of them. Yeah, 11. Vancomycin, neomycin, and metronidazole.
Oh.
Or nidazole.
Metronidazole.
Nidazole.
Yeah, nidazole.
And these deplete gram-negative, gram-positive, and anaerobic bacteria. Metronidazole is used for some fungi.
Oh,
for some parasites, protozoans.
Sure.
Right?
E. histolytica for one.
Yeah, because I remember that word.
Yeah, absolutely.
So the antimicrobial treatment goes 3 days before vaccination and then 1 day after. And they give them inactivated influenza vaccine. And they collected specimens from these people for a long time. So they could check what's going on, many different things. And the first thing, of course, is does the treatment work? So they looked at the gut microbiome and showed that it had contracted, right, in the feces. So that's fine. And then the question is, how do they respond? This is an inactivated influenza vaccine. Okay.
So inactivated virus, which means it's the standard vaccine that most of us are getting. It doesn't mean that the vaccine is not efficacious.
By the way, I did go and get vaccinated while you were gone, Dickson.
Did you?
Did. I went right.
So inactivated flu vaccine just means that they didn't get flu miss. They got the shot.
They got the shot, which is formalin inactivated. It's shot into your muscle. I got it. Yeah, at my left arm. It was sore for 24 hours, and then I'm good. So then they took these people, they immunized them, and then they take serum and look for antibody responses by neutralization of virus infectivity. Okay. Interestingly, no difference in antimicrobial treated people versus untreated people in their response to, this is a trivalent influenza vaccine, which means it has H1N1, H3N2, and influenza B in it. Okay. No difference. Wow. So there it's like.
Whereas mice, it was different.
The mice TLR5 knockout mice said there's a role for gut sensing.
You can't knock out TLR5 in humans, though.
You wouldn't be allowed to. No. You might be able to find someone, but you'd have to look through a lot of people. So, but we don't know of anyone who lacked TLR5. So then they had the idea of saying, well, why don't we look at people who don't have any memory from flu vaccine. They haven't been immunized before. That's interesting, right?
Yeah.
So the next year they took 7 people who's had very low neutralization titers before vaccination, and they had not received flu vaccine in the prior 3 influenza seasons, which is easy to do because so many people don't get the vaccine.
Right.
So you can find these people. And they did the same experiment. They gave them antimicrobial and then they immunized them. And there they found an impairment in the antibody response, but just to H1N1. Right? H1N1 impairment, not H3N2 or B. It's interesting, right? They also find that it's IgG1 that's impaired, not IgG2 or IgG3 antibodies.
So since influenza virus doesn't have any flagellae, what's the connection between TLR5 depletion.
Well, there's none in this story.
No, with the mouse model. I'm going to go back to that one for a minute. Why would that make a difference?
Well, when you take away TLR5, the mice don't respond to flagellin in their guts, say.
So?
And apparently that's not good for their responding to flu vaccine. So it's just a general, it says bacteria may be involved in responses to flu vaccine. So now we're testing it by depleting bacteria. Using
and you see this effect. And what's interesting, I mean, okay, the gut microbiome controls everything. So yeah, you perturbed it and you got a difference in the flu shot. But the specificity of this is just so remarkable.
Mm-hmm.
It's H1N1, yeah.
It's H1N1, and that's it.
And IgG1.
And IgG, yeah.
So part of it is, so obviously the memory, so in the first cohort, memory responses are not impaired, I guess, right? Because those people had previous H1N1. So what you're doing there is you have a memory. So this is more of a primary, I suppose. And then they say, what about H3N2? So I think they say later that most people have been infected with H3N2. So that's probably why. So
did they look for any IgM response?
But they did look for IgA, and that was also lower. So
no, IgM.
I heard that, but I said no, they looked for IgA.
They did not, Dickson, look for IgA.
Thank you.
Did I not say no?
No, you didn't. You just said they looked for IgA. So that would indicate that the primary response to a new antigen has yet to be investigated with regards to whether they did or not.
Well, they should look more extensively.
They should.
For other.
It's harder to look for IgM. I know it is.
But there are not a number of vaccines where people are going to be naive more easily than flu.
Yeah, but that would be the test as to whether these people who had not been vaccinated but had yet encountered the virus naturally as a way of acquiring it.
Of course, yeah.
If there's no IgM, then these people were not the right subjects to use for that experiment.
They did not look at IgM, but they did ensure that they hadn't been immunized for the prior 3 seasons.
Yeah, but that doesn't.
But I know there's some self-reporting error involved there, right?
Well, and they could have been exposed to the virus.
Exactly.
Yeah, but nevertheless, there was an effect in this study where
there was.
They didn't see one in the previous one, and I think that's quite interesting.
What do the authors say that means?
Well, the question is why, or what's the mechanism, right? And we really don't end up finding out. And there's a lot more stuff they do which doesn't really bear on this. So they actually then they look at transcriptional signatures and the effect of antimicrobial treatment on transcriptional signatures in the blood. And they come to the conclusions that there are differences and that they involve fatty acid metabolism, but it actually has nothing to do with the immune response. But the interesting thing is that the antibiotic depletion of the gut microbiome drives an inflammatory signature that looks like what happens when you get older. Okay, age-associated changes in immune responses appear to be similar to what's happening here, but that's not what influenced the antibody response. So that's kind of a side thing that they did here. It had nothing to do with it. So they really don't know why. Why not having certain bacteria in the gut influence immunization in this way. They have some ideas that maybe metabolism, because we're understanding now that the gut microbiome influences metabolism and metabolites that are produced, and maybe that's involved as well, right?
And metabolism is very much impacting immunology.
Yeah,
of course.
Right.
In particular,
so they see this pro-inflammatory profile and the look at the blood metabolites, and there's stuff going on as a result of having disrupted or as part of disrupting the intestinal microbiome.
Yeah, so they say this IgG response was associated with 2 metabolic clusters, highly enriched in fatty acid metabolism, which, as Brienne said, is emerging as an important orchestrator of immune responses. So they have to follow that up.
Would be interesting to see how germ-free animals respond to the vaccine. No bacteria.
They probably
somebody must have done that, right?
Yeah, I would think. Maybe that's some of the previously published information.
There's a lot of bacterial leftovers in their sterilized food, however, which could serve as stimulation to the immune system.
So I think if you're on antimicrobial treatment, you might want to postpone your flu vaccine, although they say that the perturbation in the gut microbiome persists for a year. It takes a long time for it to recover.
And if you're on antimicrobial treatment, you probably want to do whatever you can to avoid getting the flu.
Right.
Because you're sick with something already, right?
Right.
And if you've already had the flu vaccine, you're in the group that's going to respond just as well, whether or not you're antibiotic treated or not.
Yeah, that's true. Right. So if this is not your first time in a few years,
don't use this as an excuse not to get the flu shot.
Don't worry about it. Yep. So
I don't know. Given that I get the flu shot every year, I feel like
you're okay.
I'm okay.
You're okay. I get it every year. I'm okay. So I can take antibiotics. Yeah.
Yeah.
But if you haven't had it and I think a lot of people haven't had it for many years, and then this year TWiV makes you get a flu vaccine. If you're on antimicrobials, you might want to wait. I mean, that's not medical advice, but
true.
From what this paper says, it may not be effective, right?
Well, for H1N1.
For H1N1, yeah. And of course,
what is the flu strain?
Hey,
now?
that's a good question. Let's look. Do you know where you can look? CDC Flu View.
CDC.
Let's look and see what the predominant strain is this year. Flu activity and surveillance. Weekly influenza flu report. Here we go. Let's see the pictures. So, influenza positive tests. No, that's not what we want. We want okay, here we go. Subtyping. And now it looks like H1N1 this year is the predominant strain. Not HVN2 is being reported, but it's far smaller. And also B is as much as H1N1.
And does the current flu vaccine have H1N1 in it?
Yes, of course.
Yes.
Of course.
Well, okay.
Always H1N1, HVN2, and 2B strains is quadrivalent.
2B strains is quadrivalent now, yeah.
And how it matches this year, I don't know.
Mm-hmm.
I don't know. We could find it here in this, but we will talk about it next time. How's that? That way we don't say anything wrong, and we don't have to look it up right now. But the predominant.
We don't say anything wrong about that.
We do say other things wrong, right?
Oh, I'm famous for that one. Well, norovirus.
But you went away for a while, and you went away. Here's the last sentence of the last paragraph has a nice sentence. As antibiotics and vaccines represent two of the most widely used medical interventions, these results have important implications for clinical practice and public health.
Right.
Of course, they use this. I replaced this with these results. I don't like it if you use just this without a subject, right? Because it's not clear, not often clear what you're referring to, but people do this all the time.
Pedant.
All the time.
Pedant.
Well, it's just a writing style. I see it.
It's a writing thing.
It's a writing thing. I see it all the time. And if you're writing a paper with me, you cannot do it.
It's a dangling modifier.
Oh, it's a pronoun reference that's unclear.
I think Kathy also agrees with me on that.
Oh, yeah, of course I do.
I very much agree with you on that.
Okay. Go ahead.
I think it's a good point. Yeah.
I also did a quick search for effect of the microbiome on vaccines and found a review from August 2019. It looks like the experiment has been done, but mostly with ovalbumin and sort of vaccines like that. I don't see a specific study with the flu vaccine.
And you're talking about germ-free mice?
Yeah.
In germ-free mice, yes. Sorry.
Okay.
Right,
yeah.
Right. In this mouse.
In this mouse.
Yes.
I think it's an interesting experiment, and I can imagine we're going to see more of that, and it's going to have implications, right?
Yeah.
But I think the mechanism would be.
You might even see
curious.
a correction to the microbiome from suppositories or something like this after giving antibiotics to correct it sooner than a year.
Yeah.
Because a year is a long time to wait for this thing to correct itself.
So in yesterday's TWiV, we did a paper where so we already know that the microbiome of people with colorectal cancer is different, right? But we don't know if it's causative or caused by, right?
Sure.
What they did in this paper is they fed mice. They gave mice a fecal transplant from either people with or without CRC. And they showed that the intestinal walls become dysbiotic and they proliferate and they found that the bacteria induce methylation at the promoters of genes in the mouse intestine that are involved with carcinogenesis. So there's some regular, and they developed a blood test that you can use to screen people to see if you have these methylation changes. So it's very interesting that the effect of the gut microbiome to induce cancer may be on promoters of genes involved in cell
What's the
growth.
selective pressure for that, do you think, evolutionarily?
Well, the selective pressure, this is my idea.
Go ahead.
So we used to be hunter-gatherers, right?
We did.
And it was hard to get meat. We mostly ate roots and beans.
Berries and nuts.
Berries, right?
That's right.
And now there's too much meat around, and our gut has not evolved to deal with this properly.
Yeah, but the claim is from the evolutionists. The reason why we did evolve so fast as compared to lots of other animals is because we did eat meat.
Well,
and our brains grew faster and bigger.
And the meat is, I think, a problem. But the real question is, do animals who eat meat all the time, do they get colon cancer? That who have evolved eating meat the whole time, like lions and so forth.
Well, but their guts also evolved to eat that level of meat. I think the issue that in this hypothesis is we didn't evolve to eat quite this much meat. We also didn't really have to worry about living past age 40 because we were already grandparents by then.
This is true, but
colon cancer was just a non-issue in the hunter-gatherers.
So if you think of people living in the far north, for instance, in the polar region,
yes,
those people only eat meat because you can't grow anything there. So I wonder what their rate of colorectal cancer is.
I don't know if they die at 40, as Alan says. You're not.
No, they don't. They live long lives. They live long
Well, they do
lives.
now. Yeah, they live a long time. But
yeah, I think the hunter-gatherers didn't live
no, they were eaten by something else.
Yeah, that's right. They were eaten by someone
or
else.
they died of viral or bacterial or fungal
Eat
diseases.
and be eaten.
I'm reading this wonderful book called Sapiens.
Oh.
It's a history of...
Yes, I know it.
Right? And his theory is that the agricultural revolution was a disaster for humans because it made us all work harder and do all these terrible things that we're ending up now. Whereas he said we were hunter-gatherers 3 days a week. We'd gather food. The rest of the time we spend with our kids and chilling.
Really?
Yes and no.
He says.
And starving and
It's true. And migrating.
Well, he said
a lot of migrating.
if you could migrate and get food somewhere else,
lots
whereas
of
if you
migrating.
set up a farm, you couldn't really migrate.
Why would you want to migrate? Got my food right here.
You wouldn't have it. Well, what if you had a bad season?
No, of course. You blamed it on bad behavior. God will punish you for that. That's what happened.
Anyway, it's an interesting idea, right?
Yeah, sure.
But I mean, all the good things we have today, you wouldn't have without the ag revo. So
totally agree.
All right, our second paper is Nature Microbiology. Successive blood meals enhance virus dissemination within mosquitoes and increase transmission potential. And this is from a few places: Connecticut Agricultural Experiment Station, Yale University, Colorado State, the site of ASV this year, and Quinnipiac University, which I usually associate with Poles, right?
True. This is true.
That's true.
And Romanians.
What's that?
It was a joke. It was a bad joke. It was bad.
Oh, I get it. I'm very sorry. I'm sorry.
It's okay. You just got back from Singapore.
First author, as far as I can tell, right? There's no co-firsts. Tell me.
I think that's right.
Philip Armstrong, and the last is Doug Brackney. So this is interesting because when I was in Singapore, where they have lots of mosquitoes,
they do.
People were interested in mosquitoes there because of that.
They have to be.
But they don't have a lot of dengue there. They have, in peak years, they have 15,000 cases, they said. And they're not even that serious because they occur in older people. But they still worry about...
everybody's spending their time in air-conditioned buildings.
Correct. Exactly.
I mean, it's a built-up city, right?
Speaking of that, very interesting.
Boy, is it hot.
The mosquitoes have moved to the upper floors of these tall buildings. They told me because there's plantings in all these apartments.
Yeah.
They have outdoor, and now they have mosquitoes on every floor. If you have a 50-story building there, and they do their release on multiple floors. They can't just release on the ground because they'll never get up 50 floors, right?
That's interesting.
So they have to take the elevator released. So I didn't realize that. The other thing that came up is this multiple bites.
Yeah.
Now I know that for a mosquito to transmit a virus, it has to bite at least twice, right?
Right.
To pick it up the first time. And
No.
No? Oh, you've said this. You've agreed to this in the past.
No. The protozoans, but not viruses. Some of them are transmitted vertically, and therefore
Right,
you
so
don't
if
have to
they're
bite
vertical,
twice.
Right, but if you want to go person to person, you have to bite twice. You bite to pick up, and then you bite to deliver, right? The bite to pick up, it replicates.
If you don't have it, yes.
It spreads through the hemocoel to the salivary gland, and then the next bite, they deliver it with the saliva. Right, at least two, but apparently that you can bite more.
Multiple bites where they're biting for a different reason to transmit viruses,
well,
obviously.
Of course, Dickson.
They want to make eggs.
Yes, they don't bite to transmit virus.
They don't.
They bite.
No, they don't.
And they don't even know that they're biting to make eggs, They just bite. That's what they do.
They're hungry.
They might have an inkling about the eggs.
They don't have big enough brains to know that.
That's right. And in Singapore, people were saying that there's a theory now, and I didn't look into this, that some virus infections of mosquitoes actually cause them to bite more frequently.
But
Did you know that? We've never mentioned that.
Microbiomes controlling immunity. Come on.
So that would be another example of viruses controlling the behavior of their hosts.
For sure.
Right.
So I have to look into that if that's true. But anyway, this paper looks at this dichotomy or paradox, which is Aedes aegypti is not a very good host in the laboratory if you infect them.
Right.
But in the field that transmits viruses very well, especially in epidemics, right?
That's right.
Zika, dengue, chikungunya are the viruses that are studied in this paper that are mosquito transmitted. And so a very common experiment in the laboratory is to ask the vector competence of a mosquito. You give it a blood meal, which you take blood and you have virus in it, to which you have added virus, and then it's warmed up so the mosquitoes will drink it. And there's this apparatus, which I saw in Singapore, where you put a membrane on it. It's a cup with a membrane, and then the blood is warm, the mosquitoes come and they drink it. And then you ask, how well does the virus replicate in the mosquito, and maybe even if it's transmitted.
Right. So I was a little bit frustrated in this paper because they use these two terms and they never define them. So I looked up and found that vector competence means the ability of the arthropod to acquire, maintain, and transmit microbes.
Yes.
Right. Right. Proportion of mosquitoes that become infected and transmit, right? And I guess you could do that in mice.
But it's obvious that the mosquitoes have to allow some viral replication in order to increase the titer enough to be able to transmit, right?
So
that determines the incubation period in the mosquito, or the EIP, the extrinsic incubation period, which is
a lot here. Defined as the interval between acquisition of the infectious agent and the vector's ability to transmit it.
Mm-hmm.
Right.
And a big part of this is whether or not that virus is able to leave the gut and move to other parts of the mosquito, like dissemination, to eventually get to the salivary glands.
Right.
That's right.
Because the mosquitoes do not barf into the next bite. They you got to get out of
the virus on the skin and allow it to crawl in.
The virus gets in the gut. It replicates in the epithelium, and then becomes systemic, and then reaches the salivary glands.
I was surprised at how many times in the last semester I had to explain things like that to talk to people about why different viruses were not transmitted by mosquitoes.
Like HIV.
Like HIV.
And the mosquito initially has to take up enough virus to initiate an infection. And if the host is not viremic enough, as is the case with humans infected with West Nile virus, it will not go from person to person via mosquito.
Right.
Or horses.
Horses, triple E virus, which we talked about, won't go from horses to people. They're dead. Mammals are dead end hosts for that.
Yes.
It's the birds.
It's the boids. The boids.
Same thing with West Nile, right?
Exactly.
So here they say, there's this Aedes aegypti is really good at causing epidemics of various arboviruses, but in the lab, it's kind of a crappy, its competence is not very good.
Right.
And why is that? And they say, well, maybe because these competence assays don't allow refeeding.
But there was another one related to it that was good. That was albopictus, as I recall. Didn't they show that they had a better vector competence on a single blood meal than the Aedes aegypti?
For which virus?
For dengue, I thought.
If that was the case, that wasn't shown in this paper, and I don't remember it being referred to in this paper. So you're remembering something, Dickson.
Yeah, probably.
It's testing our memory.
No, I thought I read it in the abstract. Let's see. I'm going to
go ahead and read the abstract while we...
I'm going to do that right now.
I mean, they did look at albopictus here.
They did.
Studies.
Right.
Yeah.
All right. So
that's what
they said. So their hypothesis is that if a mosquito has a second blood meal after taking up the first virus-laden blood, that will somehow lead to more effective dissemination and transmission compared to to mosquitoes that only take one blood meal. And so they set up an experiment to test that, which is quite simple.
And I guess the logic going into this is if the mosquito's better fed, maybe it's going to be healthier, more energy available for making virus, something like that.
They actually don't say.
Yeah, they don't really spell out the logic of it. I guess the other thing is this is what mosquitoes do in the wild, so let's try simulating it in the lab.
That makes sense to me. Yeah.
The line that I'm reading in the abstract that I got this idea from, it says the competence of this species for dengue virus and chikungunya virus, as well as Aedes albopictus for Zika, that's where I got it, suggesting that this phenomenon may be more common among others than the virus vector pairings, and that A. albopictus might be a more competent vector than once thought.
Right. So a second blood meal increases competence.
Even more in albopictus than it is.
Well, not necessarily even more, but
it does.
It makes it a better vector. Let's put it.
It makes it sort of a viable vector.
Yeah. So I didn't read it anywhere else because I don't read that often, Kathy.
Yeah, vectors we haven't been thinking of as wild maybe.
So the experiment is you give a Aedes aegypti an infected blood meal, and then 3 days after you give them another blood meal but without virus.
Right.
And they're like, "Whoa, we're getting 2 meals."
Exactly right.
And then you
just like we would in the wild.
That's right.
Exactly.
And then in Singapore they had these big cages with male and female mosquitoes, and on the top was a blood container for feeding, and the females would go up, and you could see them on the wall of the cage. They were huge, all full of blood, and they would mate with the males in the cage and lay eggs. It was brilliant. So here they then measure infection of the midgut and then disseminated to leg tissue. So they take the legs off, which is something I learned in Alex Ciotta, which when I was up in Albany, it's called delegging the mosquitoes. Take off the legs and see if the virus makes it to the legs as a measure of dissemination. And they find if you give it the double-fed mosquitoes.
You should also inform
what is the hamburger with 2 layers? Is that a
Wendy's.
Double something?
It's Wendy's.
Yeah. Do they have a double name?
They might.
These are double.
Point is that the mosquito doesn't have a circulatory system.
Yeah.
The hemolymph bathes all the organs, and there's a open chambered heart that pumps the fluid around to all the organs, and including all of the legs and the antennae and everything else. So that's the reason why it can disseminate to the leg without the necessary.
Now, interestingly, if you give them blood without virus and then blood with virus, it doesn't make a difference. So they have to get the extra meal after
the virus.
And I just wonder if it's.
They claim it opens the pores in the membrane.
We're going to get to that. We'll get to it.
Sorry.
Okay, so that last experiment says that you got to infect the midgut for this enhancement. It's not just good enough to put blood in first. And this difference goes away eventually. 10 days after infection, you don't see this enhancement anymore. And they also measured virus in saliva as another indicator of dissemination, and they can see that it's increased by the second blood meal. They also do some mouse transmission. I think that's later. Okay.
Yeah, they do it for
the chick, not for
and the
Aedes,
dengue. Yeah.
They tested. So that was with Zika. Then they tested dengue and chikungunya. Same effect. More virus in the legs and saliva, and as for chick, they can measure transmission to mice, higher proportion of mosquitoes transmitting the virus to mice. They also tested albopictus,
right? Which
they make a statement that made me look up something. They say albopictus also refeeds within a single gonotrophic cycle.
I Googled the same thing.
Gonotrophic. Got a lot of hits today.
Egg making.
Is that right?
Yeah,
egg
making.
But it seems to be specific for mosquitoes, or no?
What?
I'm not gonotrophic, right? But birds are, right? Birds.
Well, maybe your wife is, but
birds
you're not.
No, we don't lay eggs, but birds are gonotrophic.
Yeah,
right?
Sure.
So are there mosquitoes that do not refeed within a single gonotrophic cycle?
I think the Anopheline mosquitoes feed only twice
in a
They only live 2 weeks. So they feed, they lay eggs, they feed, they lay eggs. That's what they do. And it takes a week in between each.
But they can transmit infections, right?
Oh, sure.
So that sounds like
malaria.
That's
2 cycles. It's 2 cycles. Yeah, it's 2 cycles.
So is there any mosquito that feeds only once?
We'd have to ask Bob Gwads that question. I don't know the answer.
You give him a call or...
Well,
you don't want to
I can't do that right now because I'm on the hotspot.
No, I'm just kidding. So, Albopictus and Aegypti, similar findings.
Yes.
Now, let me just address one issue that they say here, or one statement they make. These findings suggest that under field conditions of frequent feeding, A. albopictus are more competent and could have contributed more to transmission during Zika than previously thought. But I thought that no one really isolated Zika from A. albopictus in the field, right? Is that not a good measure of whether it's transmitted? You might miss it, right?
You'd have to ask Amy.
Bob Gwads, maybe.
Or Bob Gwads. That's right.
Anyway, so my understanding was that albopictus was shown in these kinds of experiments with a single feeding to be competent, right? But it wasn't a factor in the outbreak. So I'm not sure why they say just because we found this that it could have been I think these are interesting findings, but I'm not sure they have an impact on.
Oh, I had the impression that it meant that they thought that albopictus was not so much of a factor, but that's because they were doing things that weren't replicating field conditions. And if they did, I guess I'm not clarifying.
That was another
Sorry.
paper.
Yeah. No.
I think that the vector competence of both of these mosquitoes is probably underestimated by previous experiments, right? If you don't do a second feeding, yes. But whether it makes a difference in the field, I mean, that could explain the difference between vector competence and what you see in epidemics. But as far as I know, we did not see albopictus involved in the Zika outbreak. So even though it's better with 2 meals, there's something else limiting it, I think. That's what I'm trying to say.
Right.
Yeah.
Okay, so what's going on here? So here they say maybe this influx of energy-rich blood is helping to promote virus reproduction or replication, because actually they look at genome equivalence, and it doesn't. There's no increase when you put more blood in. Okay, so it's not just that. So then they say, well, what about the actual structure of the gut, the midgut? So you have epithelial cells, and then on top, underneath it is a extracellular matrix, the basal lamina, right? And so this is underneath the layer of epithelium in the midgut.
Correct.
Basal lamina. And they say the pore size is 10 nanometers, which, of course, is too small to allow these viruses to enter passively. But let me just say, when I teach this, when viruses infect epithelium, you get inflammation, and that makes the pores bigger.
Them up.
So just the pore size at rest doesn't really mean much because it opens up and viruses and infected cells can move through.
So in malaria, as contrast to the virus infections, the ookinete, which mates in the midgut, and the fluid in the midgut, it penetrates between the epithelial cells, but it can't go further than the basal membrane, so it just stays there, and that's where the oocysts develop. It actually causes the membrane to stretch out.
Got it. Anyway, so they look at whether the blood feeding affects the pore size. They do some electron microscopy, and indeed...
Was that 40 nanometers for most flappy
50 to 70.
50 to 70, okay.
They say that when you feed them blood, these pores get distended. Clear signs of damage by 72 hours, then it comes back, it recovers.
It may be because it's stretched during the taking of the blood.
I don't know why that would happen, frankly.
Yeah. No, that makes sense.
What, the whole gut is stretched?
Yeah, just a physical distension.
Huh.
Yeah.
I
really like this.
Having too much of your holiday meal.
Yes.
The black dot on a balloon, you blow it up and it's a giant.
I was just going to say, you blow up a balloon, the color changes.
There you go.
These mosquitoes on the side of the cage were big. You can see the blood in their guts.
They're also slow, right? So that's in real life situations. If you want to control an infection that's mosquito borne, you spray the DDT on the windowsill.
Right.
And after they take the blood meal, they're so heavily laden down, they land on that and then they die.
Of course, in a facility such as the one I saw in Singapore, mosquitoes invariably escape.
Of course.
And they have these little things that look like small tennis rackets, but they're electrified.
Right.
And they swipe them. They
Little
go,
zappers.
and
Zapettes.
we were in the room where they had all these small jars full of males for release, and
That's
there were
very funny.
there were a lot of males. And the woman who was the director, she said there are a lot of males attracted to you. And she was swabbed.
Why would you worry about males?
Those are the ones they release in this.
Males don't bite.
Well, I don't care. They don't want them to get out. Okay.
What's the downside?
What's the downside? I don't know. She was walking around swiping them. Maybe it's a thing that it's kind of fun to hear that crackling noise.
Well, it could also.
She's practicing her forehand.
It could also just be a biocontrol rule that they
what it is,
have to make their best efforts not to let anything out.
I think that, they probably don't have to.
But with so many mosquitoes in Singapore, how would you know where it came from?
Well, okay. The fact that you won't get caught doesn't mean
right.
They had extensive. They had lots of doors and screens, and everything was white and sealed.
At Notre Dame, it was. You knew when you went to George Craig's laboratory.
Oh, that's a good idea. So
you got bitten.
The actual physical distension by the blood meal, that's a great idea.
Yeah.
And that stretches the pores.
That's the gist of it.
And then, so there's no physiological chemical thing going on.
Maybe not.
No, just a stretch.
No.
Yeah.
Just you ate too much.
Yeah. What I liked about this experiment is that you can learn a lot just by looking. And they did the EM and they saw these different pores.
That's right.
It's really cool.
It's interesting that the gut tract should have a restriction on size particles for what gets into the hemocoel. It's sort of like a natural filter, a Kupfer cell almost, because the mosquito takes in a lot of stuff, right? They eat plant juices. They don't feed on blood. So they're constantly sucking plant juices all the time. There's a ton of viruses in plant juices, right? So that's maybe a natural way of preventing them becoming affected.
They also looked at the infection of the epithelium, and they could see that with a blood meal, you get higher rates of infection of the epithelium, which is a different issue because the epithelium is above the.
Right. How do they get from the epithelium?
Right. So that's another issue. Why you're getting more efficient infection of the epithelium. That's right. Not just physical distension.
Right.
And then they did some modeling to see how the second blood meal would affect transmission, and they calculated this extrinsic incubation period, which would be 8.8 days with one blood meal and 7.3 days with two, so it's shortened by roughly a day. And then they could use that to figure out the R0. And tell me, this is normally the number of people who can get infected from one person who's infected, right? But in this case, what are they do? Are they calculating in people? Are they extrapolating to people?
Because mosquitoes don't give it to each other.
No, and why would they care about mice, right? So I'm assuming this is a human. So with a single feed, 2.96, and with a double feed, 4.05.
Did they, as a control, did they show you how they fed the mosquitoes?
They have this.
I don't mean the blood meal. Did they show you how they keep them alive?
No, they have that cylinder which has got blood in it with a membrane on top, and it's heated, and that's how they feed them.
No, that's not their food.
I know it's not their food. The blood meal. That's what you asked me.
No. I'm asking you, did they show you how they fed the mosquitoes?
I don't know.
Not what they have for a feast, but what they have for their daily diet.
It might be in the extended supplementary data. I didn't read it.
No, but they feed
to clarify,
it on.
You keep saying that that's not their food. Blood is their food, but only when they're laying eggs.
Plant juices. So
they used to feed. They eat plant juice.
They used to feed the mosquitoes at Notre Dame slices of apple. And
they're giving sugar here.
Yeah. So the point is, if you did the control experiment, rather than using blood as a second meal, use apple slice.
Yeah. So mosquitoes have access to 10% sucrose. And in fact, in Singapore, they had these containers of sucrose with a gauze in it.
Of course.
Which would wick up the sucrose, and then the mosquitoes were sitting on the gauze drinking.
Exactly. So they are doing that control.
Yeah.
And in fact, most people would be.
So that probably doesn't distend them that much.
It's very interesting. Ingestion of sucrose was shown to be diverted to the mosquito crop rather than the midgut, and therefore would have no expected impact on midgut expansion and basal lamina permeability.
Oh,
well, it's a crop, Dickson.
Well, it's a region of the gut tract, but it also has another region which is very interesting. It's the diverticula. The midgut has these finger-like tubes that lead off of it, and they pack them filled with red cells. They plasmaphorese the blood, basically.
That's what my wife had removed from her colon.
A diverticulum. Yeah, but they've got...
in humans, it's not good.
No, it's not good at all. No, it's not. Diverticulosis is a bad thing.
Yeah, because the food goes in, gets inflamed.
Exactly. But in mosquitoes, that's how they sequester all the red cells. They pack them in there, and all the fluid is passed out afterwards.
I didn't know that.
Yeah.
So they normally have diverticuli for good purposes.
You show me a...
You don't have to show me.
No, it's in our textbook.
This is a podcast. You don't have to show me anything.
Right. So
I will paint you a picture.
Classic image of the mosquito excreting a clear drop.
Yeah, exactly right. Exactly right.
Because it's filtered the water out of the blood.
Exactly. Or the plasma.
So that's the story. So mosquito gets infected. You have infection of the epithelial cells. There's some dilation by inflammation. Some virus gets in. But if you have a second blood meal soon thereafter, it will help get the virus across and help it to get into peripheral tissues, salivary glands, and be transmitted. And so they say that vector competence assays may not accurately reflect that, which I would agree with. Now, Dickson, they have a concern here about using artificial blood meals to feed mosquitoes because these blood meals use anticoagulated blood, which results in lower infection rates, when you compare it to feeding on a viremic animal. And they have dealt with it by growing virus in mosquito cells where you get a higher titer. And there the glycoproteins of the virus are modified differently, and they say all these things could have an effect. So we're just telling you we don't know if they have an effect.
Right.
But we're being upfront. We do things a little differently than in the wild. Okay. I think those are good points.
Absolutely.
Right. They say that the source of virus, the virus titer, and the method of blood feeding are unlikely to alter their findings, however.
Yes.
And yet, if you did the experiment, you'd find out.
Yeah.
Right.
Well,
and then they also talk about the volume of blood that's ingested and how when there's interrupted feeding, that could result in partial engorgement. But they seem to think that, or it's not clear whether this is going to have an effect here, and they need to test that.
Why do you think that would be an interrupted blood feeding for Aedes aegypti and not for Anopheles stevensii?
I don't know, but I bet you're going to tell me.
I will. Because Anopheles stevensii is a night feeder and you're asleep and you don't know you're being bitten. Whereas during the daytime, Aedes aegypti and Aedes albopictus, they bite during the day and you can see them and you can swat them off. So they're interrupted.
So but animals...
Maybe that's why they have to feed twice, because they don't fill up their diverticula right away.
A tapir can't swat off mosquitoes. Right? A lot of animals can't swat them off. Right?
It's why horses stand head to tail.
Oh, they tail each other.
Tails wipe the flies away from the face.
So do albopictus and aegypti also bite at night as well as?
No. They're evening biters. They're like dusk.
Crepuscular?
Crepuscular.
Exactly. Crepuscular.
Lovely
That's
word.
the word I meant to.
Lovely word.
I like that one. I do too.
Huh.
Back when we were talking about whether it was mechanical distension or not, they did this experiment, which was the retrograde infection assay, where they put Zika virus intrathoracically into the mosquitoes and gave half of them the non-infectious blood meal. And then at 3 days post-inoculation and 9 days post-blood meal, they took out the midguts and looked to see whether the virus had gotten out. And the titers were significantly higher in those that were provided with the blood meal. Mm-hmm. So I just really like that retrograde infection experiment, and it really does point to the distension being the mechanism.
Mm-hmm.
So the last sentence of the paper is, the findings emphasize the importance of considering feeding behavioral traits when performing vector competence studies.
Yeah.
Past studies may underestimate the risks of arbo transmission by measuring vector competence after only a single infectious blood meal. So I guess the bottom line is you should give them 2 blood meals when you're measuring vector competence. I wonder if anyone's going to actually do that, the people who work on this.
Maybe if they listen to us, they will.
It doesn't seem that
or they
hard,
read this
right?
paper.
Right.
No,
Read the
it
paper,
doesn't seem very hard, yeah.
So one of the podcasts that's going to come out soon is one we did in Galveston, where we talked with a bunch of arbovirologists. But they didn't bring this up at all. It'd be interesting to see their thoughts. Anyway.
I think that one thing that it brought up for me was sort of the importance of thinking about biology in a number of different scales. Because usually when I think about, viruses, I have not thought frequently about how many times a mosquito bites in its lifetime.
Right.
But then when reading this paper, I went down a Google rabbit hole to learn more about that. And it sort of made me excited to be in a general biology department and to realize how all of those things go together.
Exactly.
On so many levels, reductionism in science is always an issue, right? So I spent most of my career infecting plates of cells with viruses, right? Thinking that this is anything like what really happens.
Right.
And, of course, some aspects may be, but there are many that are not, right? But that's what we do.
Sure.
And it's not just virology, but it's all kinds of science.
It is.
You have a simple system you develop.
Lots of people in my field, at least for the Trichinella field and other parasite fields as well, used highly inbred strains of mice to do their experiments. And nowadays they're encouraging you to use outbred mice to show what the normal pattern is like. And it's quite a reversal of fortune.
Okay, there we go. Here we have a few emails now. We have lots piled up, but we will get through them. First one is from Ryan, who sends us a series of articles in the Daily Mail about Andrew Wakefield, who has apparently gotten called out for living a luxurious lifestyle, which can only occur when you make lots of money promoting anti-vaccination. This is at a time when Wakefield has also been accused of trying to influence the 2020 elections here in the U.S. by setting up super PACs and lobbying offices at state capitals in Washington, D.C. to prevent California vaccine laws SB 276 and 277 from going nationwide in the USA.
These are criminals.
And apparently he's dating supermodel Ella McPherson.
Criminal.
Is what some of these articles are about.
He should be dating Jenny McCarthy.
Jenny
Right. Wow. And from Carol, Dickson, can you take the next one from Carol?
Sure. Carol writes, Dear TWiV, if you're wondering why people distrust modern medicine, here are two articles. And she lists one from the LA Times, which is a science story about the opioid trial settlement challenges, as to why it's been going on so long without anybody noticing, except the poor people who have lost people because of that. And then another one is in the Washington Post, which is about a lawyer that's suing the nation's largest drug companies over the opioid crisis as well. The articles are about the fallout from the opioid abuse scandal. There have been protests and lawsuits around the country over how doctors and pharmacists have failed to stop the opioid abuse around the nation. I do not know if this opioid scandal directly caused voters to support the anti-vax movement, though, but we have one factor.
Well, general distrust is always a problem.
It is, isn't it? It is. Definitely.
Yeah, I mean, this is, to me, this points to a failure in a system of unbridled profit. It's not so much the drug industry as it is, just the fundamental system of profit at all cost, and that's our focus. And then you get the Sacklers who profited off selling huge amounts of addictive drugs, and
it's all about money. It's all about profits. I've thought the same thing now for many years. It's very sad. What are you going to do? Amongst the profit mongers, there are honest people who are actually nice. Kathy, can you take the next one?
Sure. Suellen writes, I love the discussion of Eastern equine encephalitis in TWiV 567. As a horse owner, let me add a few facts. 1. Responsible horse owners vaccinate at least once a year for Triple E as well as for West Nile. Here in Georgia, as across most of the South, we vaccinate our horses twice a year, spring and fall, because we have a longer mosquito season. 2. Brianne is right. Dickson was maybe a bit right. New Jersey is an important horse state, not just because of racing, but because of all the sport horses in the state. As Brianne mentioned, the U.S. equestrian team is based at Gladstone, And since an Olympic-level horse is worth upwards of $500,000, you can bet they get vaccinated regularly. 3, I was really glad to hear that mammals are a dead-end host for TWiV because I've actually had people ask me if they can get TWiV from a horse. I can now point them to the discussion on TWiV as proof that no, you can't get TWiV from your horse, even if your horse isn't vaccinated. 4, horses are herd animals, so one horse by itself can become stressed or, yes, depressed. So kudos to Sweden for requiring horse owners to have at least 2. By the way, a friend tells me they also require guinea pig owners to have at least 2 for the same reason. 5, yes, horses get depressed, especially when they are not well. I realize it's hard for non-horse people to imagine this, but horses do express emotion. A depressed horse will hang its head, usually face a corner of the pasture or stall, and may stop eating. And a good horse owner can tell when his or her horse is depressed. On a non-horsey note, I really appreciated both Brianne's and Rich's non-technical explanations of the budding process in HIV and of what the paper on HIV was all about. For those of us who enjoy the show but don't have PhDs in virology, it's much easier to follow the technical details of the paper when we have that 30,000-foot view of what the paper is about. And sorry to be so long-winded, but I don't write often. I wanted to add one more thing. A book I read recently that was an enormous help to me in understanding the basic pieces of the immune system. I love your immune podcast, but it's often way over my head. Now I have at least some basic knowledge that I hope will help me follow the discussions. The book is An Elegant Defense by Matt Richter, and although it tended to give a bit too much detail on the patients he followed, it also managed to give a really excellent overview of how the immune system works, so I recommend it to immune system newbies like me. Keep up the great work. I love the show. Don't stop discussing the weather, and don't change the titles. I love the puns. Scientists can be so clever. LOL.
So, you want me to tell the joke or not?
Go ahead, tell your joke.
So it's a joke that sort of backs up her statement about horses getting depressed. A horse walks into a bar, and the bartender looks up, pauses a moment, and says, "Why the long face?" Now that's... Kathy, you didn't like that joke when I told it before we went on the air.
I'm just going to have to ruminate on it. For a while.
Oh, lordy.
Brienne, do you know this book, Elegant Defense?
I do. It's a very good book. I actually really agree with exactly how she describes it. Sometimes it goes a little much into the patients, but the description of the immune system is fantastic.
Great.
So just horses and guinea pigs get depressed? Nothing else?
There are no road signs on my highway, so I don't know anything about anything other than the equestrian team.
Alan, can you take the next one, please?
Yeah, I'll take the next 2 because the next one is just a link. Bill writes, there may be some use for this site to make assorted capsids, molecules, etc. And he links to a Hackaday post about looks like a kind of an advanced Tinker Toys type of system that you can put together modular items from. It's kind of cool.
Always looking for ways to make virus models, right, Dickson?
Yes.
Right, Vincent.
We don't need any more ways to make more viruses. They do fine.
No, they do fine.
No, but lots of models for class.
Lots of models, yes, that's good. So Matthew writes, Dear Vincent and the gang, I have some slides on the founding fathers of virology that I use in my teaching. Currently, I go through Ivanovsky, Beyerink, Bang, Ellerman, Rouse, Twort, Dirrell, Stope, and Bittner. No Racaniello yet, but it's a posthumous thing, so you'll have to wait.
Is it Stope or Shope? I bet it's Shope.
I think it's probably Shope, yeah.
Yeah, I think it's a typo.
Processing. Okay. I wondered if there was anything that should be on this list but isn't, anyone who should be on this list but isn't, or maybe should be replaced in your esteemed judgment. Also, I'd love to follow up this slide with a few on the diva daughters of virology or something like that to highlight some of the amazing women virologists out there. And wondered if you could each suggest a few in addition to Kathy and Brianne, of course. But again, I assume
they're still alive,
right?
Yeah.
And Matt is University of Lincoln in Lincolnshire, UK.
Right.
I would suggest that we get rid of the fathers stuff and just say founders of virology, right? That's what. And don't put the women on a separate slide. Just put them all on one, right?
Yeah.
Everybody on the same slide. Completely.
I mean, we've done that with our parasite heroes now. So.
And I like your Distrust, Vincent, that you linked to.
Yeah, this is in 2011. I had done this, and actually we did some TWiVs where we mentioned some of these people. Go ahead, Kathy. Why don't you talk first?
Well, when I then read this blog post about 10 seminal virologists that David Baltimore and Vincent picked, they're all men, and I'm hard-pressed to think of women. But I was preparing my lecture for undergraduate virology, and for the first time in a long time, I'm teaching the first lecture, and so I was looking up things about tulip breaking virus, or I guess its other name is tulip mosaic virus. Anyway, it turns out that a woman was the one who discovered that a virus was causing this. Her name is Dorothy Kaley, and she discovered this in 1928. Is this a story I have ever heard before about a woman? No.
That's great.
But I'm going to tell people about it now. And I just looked her up in Wikipedia. She was a mycologist. She was a founding member of the Genetical Society, and mostly a mycologist. She did volunteer work at the John Innes Center, and she was forced to work in the attic of the manor house before the laboratories were built. Now, I don't know whether that was because everybody was or just she was, but anyway. So I now have at least one woman I can talk about.
I mean, if it's restricted to the dead, then it's harder. But I think you have Yvonne Barr, right? Epstein-Barr virus is and then Françoise Barré-Sinoussi for HIV, right? Mm-hmm. Those would be. So I don't know why they have to be. I don't think they have to be dead. I would say, and they don't have to be old either, right? It could be any time in the spectrum of history. So I think those. So
then we could include Joan Stites.
Yes. Yeah, sure.
Mm-hmm. And others.
Yeah, I always cover the Marilyn Rusink experiments with the plant fungus virus example.
Yeah,
so I would put her there.
Although this tulip thing is, you could argue that that's a first example of a good virus, although it's good for us, not for the tulips, I guess.
Well, right.
Mm-hmm. Right. Mm-hmm.
Yeah, I think
and she fits the criteria because she died in 1955.
Yeah. So David, sorry, go ahead.
I was going to say, you could also put in Rosalind Franklin, given some of the things she did with virus structure.
Yeah. It was,
and her work with Aaron Klug.
And also I was thinking of someone in the polio field who, I think what's the guy I interviewed in Sweden?
Norby?
Norby. In one of his books talks about a woman who was working on polio at Hopkins and would have probably made the first vaccine hadn't she decided to leave the field, right?
I just listened to that episode.
And that would have been cool. Anyway, David's list included Jenner, Bayerink, Rouse, Enders, Lvoff, Stanley, Klug, Delbecco, Delbruck, Temin, Baltimore, and Hilleman. He put himself on his list. I think that's good. So I would not put Stanley in because what I learned from Erling Norby is that he wasn't very good, and some other people as well. He, thought viruses were infectious proteins, basically, and ignored the 5% phosphate.
Alfred Mirsky did too.
Anyway, my list was Bayerink, Darrell, Tyler for the first attenuated vaccine, Max Delbruck, Lvoff, Hershey, Chase, Enders, Klug, Baltimore, and Doherty. But I think we should add some of the people we've mentioned also. Anyway, these are ideas for Matt.
So a lot of people have used viruses as tools to get to the molecular biology of DNA and stuff like this also. Yeah. So are those virologists or are they using them as tools?
It's a tough question, right? I mean, can you name someone in that vein that you're thinking?
Well, I mean, anybody associated with the molecular structure of how DNA replicates. Don't you have to use shuttle vectors and mutations in the various genes that you put them in with phage viruses and stuff? No? Or am I imagining that too, Kathy?
I think the point here is it's seminal. They made important contributions to a broad understanding of viruses. So if that's the case, if they figured out polymerases, why not?
Yeah.
They could be included there.
Yeah.
Anyway, there's some ideas for you, Matt. One more. From Theodore, greetings from sunny Florida. This recently published open access article in Science provides another strong reason to have all children vaccinated for measles. Thank you all for your continued dedication to these fabulous podcasts. Ted is at the College of Dental Medicine in Nova Southeastern University of Florida. And he is referring to one of the papers we talked about on a recent episode of Immune, number 26, Measles Erases Immune Memory. It's very cool.
His Darwin quote is one of my all-time favorites.
It is not the strongest of the species that survive, nor the most intelligent, but the one most responsive to change.
Exactly right.
Are humans being responsive to change, Dickson?
Responsive or responsible?
We're certainly causing it.
Right. Go to Australia right now and you can tell for yourself.
In reading this Sapiens book, I have to tell you something else. He talks about how, the first humans came to Australia and totally trashed the place in the first 50 years. Totally trashed. All the huge mammals that used to live there, gone.
That's right.
Terrible.
And some of the birds.
There were things the size of dinosaurs that were mammals. They're all gone now. My gosh, it's too bad.
Megafauna.
And they would, the humans would go up and spear them, and they're like, huh?
Exactly.
They're not used to it.
Yeah.
That's right.
Wow. Okay, let's do some picks.
Yes.
Hey.
Oh, boy, we have more mails here. Brianne, what do you have for us?
So I found an Etsy site that sold a retrovirus cookie cutter. They actually have other bacteria and virus-related cookie cutters as well. So they've got a phage, they've got an adenovirus, they've got rabies, they've got antibodies, got all sorts of medicine and science types of things. And
I have these.
Yeah, my sister and I made them over Christmas break, and we really enjoyed having virus and antibody cookies. I don't know about the rest of our family, but we thought they were great.
This is awesome.
Yeah,
these are great.
And they work really well.
Yeah, my student gave me these when she graduated, and I tweeted my picture of the cookies, and it had a lot of retweets and likes. The most tweeted thing I've ever produced.
It's not surprising.
Yeah,
This is great, though. This is great.
Yeah, they are really good.
So what viruses do they have? I'm looking at retrovirus.
They have a retrovirus, they have a phage, they have an adenovirus, and they have rabies.
And then they have some other stuff too, like GATNC molecular models of a pipette.
Yeah,
they have a 96 well plate if you need a plate.
Yeah.
I was given
that's great.
DNA and a brain and a mouse, and they will custom make you cookie cutters if you want.
Yeah,
because I assume these are 3D printed.
Yeah.
I just sent some of our cookies to the Slack channel.
I love this Bakerology. Wonderful.
Yes,
really nice. I got to get some of these.
I'm a fan. I think a lot of my students have been buying from them. I showed my students at the end of the semester.
I like the.
We all had a good time.
Floppy drive. That's cool. Very cool stuff. All right, Alan, what do you have for us?
I have a Jack London short story, which this is going to seem like a really weird pick. What the heck is this got to do with science or virology? But it's a short story, so you can read it in, 30 minutes or so. And it is a classic by Jack London from, I think it was originally published in South Sea Tales. And just have a read through it. There's a reference to events that are timely, I think, given what happened a century ago and the story that he's talking about, and given what's happening today in Samoa.
And it's open access.
It's open because it's a century old. It's open access. I pointed to a particular site where you can find the plain text of it, but you can find a dozen different versions of it.
Cool. Well, I've never read this, so
it is now. If you haven't read much early 20th century literature, you may be shocked at the lack of political correctness in it. But the point he's making is actually fairly progressive if you read the story for its meaning.
Oh, we've gone full circle in political correctness or lack thereof. Huh?
Yes.
Cool, Dickson. What do you have?
I have something that's completely way off the subject, but something that's fascinating to me, and I hope it is to you also. And it's about body size. And as I grow older, my body seems to be increasing in size. So maybe that's why I was attracted to this article. But what this says is the title is fascinating. It says, Why Whales Are Big But Not Bigger. Physiological Drivers and Ecological Limits in the Age of Ocean Giants. And there's a lot of authors on this. The graphics are spectacular because you don't have to know you don't have to read the article in order to understand what's going on. But the stomach contents of sperm whales and the ability to detect whether or not a sperm whale has made a catch or not using radio telemetry from suction-cupped video cameras is incredible. I love that. I mean, I just love that. But if you turn to the last graphic, which is figure number 4, it's a wonderful summarization of an entire article in one picture. And this is an Edward Tufte that I've never seen.
Oh, is it really by Tufte?
No, it's not, but it's something that he would just say, this is spectacular. In the style of. This is the connection between art and science.
It's a summary, not a summarization. That would be making it warm.
No, okay, fine. So this is what I mean by art and science. You put art and science together and you get one of these.
That's cool.
They're marvelous. And this raises another question, of course, because every big animal on the planet right now is a filter feeder or a plant eater. If you go back 100 million years ago, where much larger animals existed in the ocean, they were all predators, every one of them. Mosasaurus, all of those gigantic things.
Those were the biggest?
They were the biggest. They're much bigger. I mean, remember the Jurassic Park scene, the latest one, where it's like a theme park and you go and you're sitting in a big arena. You're like watching the killer whales jump up with the people on their back. They hang a great white shark over this tank and out of the water comes a mosasaur that's like 50 times longer than the goddamn shark. And that's how big the animal actually was.
Why don't they exist anymore?
That's a very
for them to eat.
This probably addresses that issue.
So right now, the blue whale is the biggest living thing on Earth.
Yeah, it is, and it's a filter feeder from start to finish.
It's the biggest animal
that ever lived.
I think it's a fungus.
Biggest living thing.
It's the biggest living
fungus, but there's some debate about what it is.
That's right. And the biggest living land animal, of course, is the elephant, and that's an obligate plant eater.
But it wasn't always the biggest, right? Or was it?
You mean elephants?
Yeah, were some of these mammals in Australia?
Oh no! If you go back, I see what
dinosaurs were much bigger.
Dinosaurs were huge.
Yeah, right. Yeah. That's true.
And they were all the biggest ones were plant eaters as well. So it's a available food source that determines size, basically, and how you access that food.
Of course, we've screwed everything up now.
Of course we have. Imagine all these poor filter feeding animals when they go through the gyres with all the plastic in it.
Every time you say filter feeding, I think of gefilte. Sorry.
That's how you keep your aquarium clean. You get a gefilte fish to come in and clean it up.
Thank you, Dickson.
You're welcome.
Kathy, what do you have?
I picked Optical Illusion. It's from a contest about the best illusions of 2019. So I don't know if this is the winner, but it's a really cool one. And you just watch this thing and you try and figure out whether it's rotating on the vertical axis or the horizontal axis. And then they show you each axis and you can see. And then after that, it leads into, a rabbit hole of YouTubes of optical illusions. But this one was just really cool.
This neat. 2 million views. Wow.
Yeah.
So tell me why, it's got 12,000 likes, thumbs up.
It's got great music.
And 300
also.
thumbs down. Why would you give this a thumb down at all? I don't understand. People just want to be negative, right?
I think so. I think
so. Sad.
You got it right.
Yeah.
It's so sad.
That's true.
All right. My book is rather negative. So I was at our faculty holiday thing weeks ago, and I was complaining. I shouldn't say this, but I'm looking around the room. Most of the people are administrators. I don't know why, but it's way more than I remember. And so Matteo Perotto, a faculty member, he ran in his office and got this book, The Fall of the Faculty, The Rise of the All-Administrative University and Why It Matters. It's really good by Benjamin Ginsberg. Let me just read one paragraph. Until very recently, American universities were led mainly by their faculties, which viewed intellectual production and pedagogy as the core missions of higher education. And in fact, when I entered here, that's what I thought a university was, for sure. We taught. Today, as Ben Ginsberg warns in this eye-opening book, deanlets, administrators and staffers, often without serious academic backgrounds or experience, are setting the educational agenda. And that's what this book is about. It's great. It's pretty short. So any of you in the university setting, or maybe if you're just interested, this is what we have become. We're run
so
by
it's basically tracking the effect of Parkinson's law on universities.
Exactly.
Yeah.
You got it.
Yep. I mean, really, I came here to spread information, and we're doing that, but I just feel that it's not what. And today, in the way in, I see billboards on highways every day advertising universities.
Yep.
And I think that's so absurd. If you're good, people will come. You don't need to advertise.
No, well,
but you know what they're doing?
marketing.
Your next career begins at this university.
South New Hampshire
And you
University.
can bet that the big ones are not advertising there.
No, they're
right? It's mostly the smaller ones. Wow.
Phoenix, Phoenix University?
Oh, the online university.
Yeah, you know what the only building that they have there is there? It's the football stadium.
They have a football
They
team?
do.
They do.
Oh, wow.
That's ridiculous.
That's
Nobody
their only
goes
building.
there.
They do. They hold a lot of football games there. They rent it out.
Oh, my gosh. The world is just crazy.
It's sad. It's very sad. But, you can see through a lot of that crap.
We have a
I've
list.
heard the word deanlet before. I wonder if this is where it came from.
Oh, yeah.
Oh, yeah. I think
heard
it
it.
existed before
Probably
this book.
before the book, but yeah, deanlet, I've heard. Yeah.
Elvin Cabot was one of our great members, and a lot of people wouldn't agree with that because of his political leanings, but nonetheless, he had great lines, and one of them was,
I wanted you to. What is that?
what's the difference between a mouse and a rat? What is the definition of a mouse and a rat? A mouse is an associate dean in training to become a dean.
I was trying to think of that recently.
A mouse is in training to become, that's right.
A mouse is
An associate dean is a mouse in training to become a rat.
rat. Okay.
That's the way that reads. That's
Good thing you're not employed here any longer.
that's a damn good thing.
But actually, you can say what you want at universities for the most part. Still, at least, for the most part, not exclusively. We have a listener pick from Suellen. Suellen writes often to TWiV. So welcome to TWiV. Found a good podcast. Thought I'd share it with you all. It's from New Hampshire Public Radio. Its name is Patient Zero. It's a history of the discovery of Lyme disease.
Oh, yeah.
There are 7 episodes so far, and I've only listened to the first one, but it seems like it's got good science. The people they interview are physicians and others involved in the search for the source of the disease. So check it out. Suellen in Roswell, Georgia, where it's cold. Only going to hit the low 50s today. Oh, yeah.
Jeez. Get out that overcoat.
Speaking of podcasts, now, I don't know how many people are still listening, but I should have put this up front, and maybe I should splice it up front. So we're going to change the RSS feed of this podcast in the next week or so. You don't need to know what that means. It shouldn't matter to most of you. But if you suddenly no longer have TWiV on your phone or tablet or wherever you listen, just go resubscribe, okay? And that may be a rare occurrence of what we're going to do.
Doesn't RSS stand for really stupid simple?
Really simple syndication.
It's really simple syndication, but KISS is what you're thinking of. Keep it simple, stupid, right?
That's right.
Yeah, it's not the same. Okay. All right, TWiV is at microbe.tv/TWiV. That's our web page where you can find the show notes and so forth and links. And if you want to send questions or comments, send them to TWiV at microbe.tv. And many of you have decided to support us in the last few weeks. At the end of the year, people want to make their donations to get their deductions for the year. So we appreciate your support very much.
Thank you.
And for those of you who would like to support us in the coming year, all the way up to 30/30, right?
Yeah.
Yes, I also very much recommend the Cafe Press TWiV hoodie that I am currently wearing in my cold office.
Very nice. Yes, over at cafepress.com/TWiV, you can buy all sorts of things, not just for TWiV, but all the other podcasts. And if you are a supporter, depending on what level, you get some of those items sent to you for free, like a mug or a t-shirt and so forth. Microbe.tv/contribute, Patreon, PayPal, boluses of money are welcome by check or PayPal as well. We're very welcome. We use it to pay our expenses and to perhaps try and do a remote episode now and then. So
we also accept briefcases of small bills.
Hopefully with consecutive numbers on them.
All the traveling I do, by the way, is largely supported by people who invite us. We don't use TWiV funds for that because we don't have enough, frankly. The money we get in wouldn't, well, the plane ticket to Singapore was $1,700. It's a lot of money. And I'm very grateful to the local meeting organizers who paid for that. But I hope you will enjoy the 4 podcasts that came out of that. Dickson Despommier, Racaniello.org, thelivingriver.com. Thank you, Dickson.
This was a lot of fun, as Kathy would say. And I would say that double.
Why don't you come up with something else?
I had a great time.
Okay. Everyone has something slightly different.
It was fun.
Welcome back.
Thank you.
Hope to see you again.
You will. Next Friday.
Brienne Barker is at Drew University in Madison, New Jersey on Twitter. She is BioProf Barker. Thanks, Brienne.
Thanks. It was great to be here. I'm glad that we're back for the new year.
Are you having TWiV withdrawal?
A little bit, yeah.
Kathy Spindler's at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This was a lot of fun.
Alan Dove is at turbidplaque.com. Over on Twitter, he's Alan Dove. Thank you, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank ASV and ASM for their support of TWiV and Ronald Jenkins for the music. This episode of TWiV was recorded, edited, and posted by me, Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.